Thanks for the invitation from Helen and the organizers. I'm very glad and honored to give a presentation on our recent work on imaging BBB water exchange with diffusion-weighted arterial spin labeling. As you know, the blood-brain barrier, BBB, plays a critical role in the exchange of nutrients between the blood and the brain parenchyma, and also preventing toxic material to reach the brain. It also was involved, in the recently proposed glymphatic system that, hypothesized to play the role in the waste clearance of the brain. So the standard way, or the gold standard for measuring BBB function or permeability is dynamic contrast enhanced MRI, or DCE MRI. So here's the protocol used at our institution, made by Dr. Rokowicz's group at the USC. It involves almost 17-minute scan with the injection of gadolinium-based contrast agent, and the in-plane resolution chrono is really high because we're targeting hippocampus, can reach millimeter, even sub-millimeter. So, with this protocol, this group has published a series of high-profile papers from Neuron in 2015 to Nature this year. Basically, the finding is really K-trans in the hippocampus will increase with aging, increase with ApoE genotype 4, and also CDR, which is the mild cognitive impairment. This just shows you the plots of increased K-trans with ApoE and the CDR. For mapping subtle BBB permeability change or functional change in cerebral small vessel disease, I think DCE was challenging because it takes a long time, on average 15 to 20 minutes, so that could involve motion and the scanner drift. And also the protocol has been really heterogeneous across different groups, as well as analysis methods. So there has been some consensus effort try to standardize this protocol, and such as the Hannes group in Europe. In the past 5 years, there has been a trend try to assess water exchange across the BBB by our group as well as several other groups. Why you want to do that is because, first, water molecule is really small, right? So if you look at the molecular weight, the water is about only 1/10 of glucose and about 1/50 of the gadolinium-based contrast agent. So it's very sensitive to subtle BBB changes. And it also, the water exchange is tightly related to this glymphatic system, as we talk about. It was hypothesized that, the water in perivascular space of the arteries or arterioles, they were pumped into the brain parenchyma by this aquaporin 4 water channel distributed on the endofoot of the astrocytes. And they wash through the brain, carry away the waste, including amyloid beta 42, and they get drained to the perivascular space of the veins and venules. So this is hypothesized as the brain waste clearance system. So water exchange actually happens at a very high rate in a normal brain. With aging, the system kind of, deteriorates and get dysfunctional, get clogged by wastes. And then in the end stage with Alzheimer's disease or other dementia, you will see the whole system just collapse. So how do we measure water exchange? So of course, our group has been working a long time in arterial spin labeling. So the arterial spin labeling use magnetic labeled water molecule in the arteries as endogenous tracer. So when it flows into the capillary, it will actually extract and exchange across the BBB into the brain tissue. There's a small fraction, maybe 5%, will be outflow into the veins. So if we ignore this component, so the labeled blood really has two components, one in capillary, one in brain. So this is just the simulation by my colleague Keith and Lawrence. So the black curve is the total labeled signal, ASL signal, and the blue is the brain tissue signal, and the red is the capillary signal. So the goal is try to differentiate these two components so we can estimate the exchange rate. However, these two components, they have very close T1 values. So if you just based on T1 value, if you only assume a single component, you actually get very close curves, so which is difficult. So our method has been really tried to use diffusion to differentiate these two component cell signal. For example, this is our experiment in human subjects showing diffusion-weighted ASL signal as a function of B value and acquired at 3 post-labeling delay from short to long. So as you can see, there's a clear fast decaying component which represents the capillary, and the slow decaying component which represents the tissue. And this fast component would the fraction would decrease with prolonged post-labeling delay, which means more blood exchange into the tissue. And the separation is around 50, the p-value of 50. And we actually replicated this finding in primates. This is working with Monica Wei at MGH, and independently in rats by Dr. Tim Dong's group. So very interesting, you will see the separation of the two components. The p-value gets smaller in rats, increased to maybe around 25 in primates, but in humans it's actually 50. So there's some kind of species difference in terms of the BBB microstructure. But nevertheless, we can reliably observe this phenomena across species. And then you can do a bi-exponential model to separate them. And of course, in a real experiment, you wouldn't be able to get so many points. But my colleague Keith and Lawrence has developed a two-step estimate model, so we can shorten the scan time. And our methods has been independently validated by Dr. Tim Dong's group. They inject mannitol in the arteries of the rats, and you will see on the lesion side, this is Evans-Brook historic validation, and we will see actually KW decrease with injection of mannitol. This is same group. They used the ischemic and the reperfusion model to damage BBB. In using our methods, so this A2 represents the fraction of labeled blood in tissue, which means this reduced A2 means reduced KW. So on the other hand, this is increased K-trans and Evans-Brook validation. So basically, there are more animal experiments suggesting reduced KW with BBB damage. So in humans, it has been a long journey. The first paper published 2007, because ASL signal is small and the diffusion with the ASL signal is even smaller, right? Until 2-3 years ago, I have a very talented student, Xinwen Shao. He developed this diffusion prepared 3D grays Picasso, which increased the sensitivity and reliability. This just shows you the KW map actually, we recruited community Latino subjects, average age 70. and they participate repeated scan about six weeks apart. So this is not your standard regular, lab technician student subjects. So we achieved the ICC of 0.75, which is very good. And in this cohort, we also see KW difference between diabetic, hypercholesterol, and also YMAD hyperintensity and cognition. So this group is very interesting. We actually see KW increase with diabetes and also my cognitive impairment. This is the study we published this year with Dr. Brian Gold at Kentucky. So in 38 healthy aged subjects, they're cognitively normal, and they all have CSF A beta measurement, and we found in the whole brain and most cortical regions that KW was correlated with CSF A beta 42. So high KW is higher CSF A beta, but lower brain A beta. So this is matched the theory of high water exchange is clearance of the A beta 42. And we only, didn't see this correlation significant in the medial temporal lobe. This just shows you a representative image of KW increase in 3 subjects, and they all have increase in CSF A beta 42. But the CBF is actually not necessarily correlated with A beta 42. And also KW was correlated with memory composite score. And the trend with executive function composite score, although it's not significant. Other groups using similar approach. This is Jack Wells group in UCL. They used a knockout ApoE for aquaporin for knockout mice compared to wild type. They use multi-echo ASL technique, which is similar to the diffusion, but based on the T2 difference between the 2 compartments. They measure increased water exchange time in aquaporin for knockout mice. The water exchange time inverse is KW. So increased water exchange time is reduced KW, which matches our human study. They didn't see any significant difference in ATT CBF and the diffusion coefficient between wild type and aquaporin for knockout mice. We were just putting data together for ISMI deadline. Today is actually ISMI deadline. You can see, across multiple study, we have about 100 subjects. CBF decrease, ATT increase with age, which is well documented in literature. Also, we saw KW decrease with aging. So this trend, again, is consistent with our observation of KW. Increased KW is correlated with higher clearance, of the waste. We were recently we acquired the data in CADASIL patients in China. So CADASIL, is a genetic form of small vessel disease. So it's related to damage of the smooth muscles and also the parasites. So they don't have the confounding effects of amyloid related to the dementia. So in 27 CADASIL patients versus 28 controls, we saw significant reduction of KW, but CBF actually was not significantly different, although it's decreased a little bit. Again, they suggest damage of BBB is related to reduced KW. And we also have a small cohort of even rare form of genetic small vessel disease called CADASIL. So this is actually have a little bit more severe form of in terms of symptom dementia, vascular dementia compared to CADASIL. So in these 10 subjects with 7 controls, we also see consistent reduction of KW in gray matter, white matter, most of the cortical region, as well as putamen, hippocampus. In this cohort, actually, CBF was also decreased. Furthermore, we compared the KW versus the K-trans in DCE MRI, again in our Latino cohort, 16-aged Latino cohort. So this is the KW K-trans map acquired in coronal high-resolution in plane. This is our actual, resolution-wise, KW is of course lower. And this shows you we observed correlation between KW and the K-trans in 3 regions. One is the MC perforated territory, subcortical, caudate, nucleus, as well as deep white matter. Nevertheless, K-trans was able to detect increased K-trans in white matter hyperintensity penumbra, which is the area surrounding the white matter hyperintensity, compared to white matter hyperintensity itself, while KW was not able to detect significant difference. So this mainly we saw the attribute to limited spatial resolution. To summarize, as you can see, it is feasible to map BBB water exchange with improved pulse sequence. And it seems the majority of our data suggesting decreasing KW with aging, cognitive design, and the small vessel disease. Of course, we do see occasionally increased KW, especially in our Latino cohort. And there's evidence suggesting KW may be related to glymphatic flow, especially aquaporin 4 function and the brain waste clearance. There are many applications, small vessel disease, aging, dementia, sleep. Sleep is very interesting. Dr. Chi's expertise area, and also glymphatic function. Of course, we also want to understand the KW more, hopefully with, preclinical animal models. With this, I would like to thank you, especially my talented student and scientist, Xinhong Shao, and the many collaborators around the world, and the funding support from NIH. Thank you very much.